Good morning, dear participants. We welcome you to the session 1 of day 4 in the STTP program. We would like to introduce the speaker of today's session, Dr.K.M. Gothandam, Professor, School of Biosciences and Technology, Vellore Institute of Technology. He has completed his PhD in Genetics and Plant Breeding, Bharatiyar University, Coimbatore. He also has filed a patent in profile-rich protein gene from Oryza sativa, inducing cold tolerance in plants. He has served as principal investigator and co-PI in many funded projects, which are funded by uh, DST, DBT, and CSIR. He is also awarded many awards such as a Fellow Biotech Research Society of India, DST Young Scientist Award, Fellow Royal Society of Biology. His research interests include functional genomics, plant secondary metabolites, microbial metabolites and enzymes, and cancer biology. He has also visited many universities abroad such as Boston University, Cambridge University, University of Miyazaki, Korea University, and Kuwait Institute of Scientific Research. He has completed guiding 11 PhD students and 6 are ongoing. His publications include more than 130 publications in peer-reviewed journals, including national and international status, with a cumulative impact factor of 230. And he has also published many book chapters of around 15 and edited books of around 7. With a short introduction, we welcome Dr. K.M. Gothandam on behalf of the College Management, Department of Biotechnology, HOD, staff and students. Over to you, sir.
Yeah, thank you, Dr. Ajita. And I also thank the HOD and other organizers for inviting me to deliver a talk in the AICTE sponsored 1 week short term training program on startup driven by innovation IoT for sustainable agri-tech. So thank you for inviting me. It's my pleasure to be part of this event. And also once again, thank for inviting to deliver the talk. So just I'll share my slide. Yeah, so good morning to one and all. Today I'm going to share some of our research work, what we carried out in our lab. So as mentioned in the introduction, so we are working in 3-4 main different areas. One is like plant functional genomics and plant biotechnology, and other area is plant and microbial metabolites, and also we are looking for metabolites for mainly for cancer prevention and treatment. But today I'm going to talk about the plant biotechnology, especially what we're doing in algae. So what we are working for past few years in the algal biotechnology. So in this specific topic, we can say that what are the various approaches to accumulate carotenoids in microalgae. So especially we are mainly working in microalgae, Dunlady salina and Chlorella pyrenosa, and also we are working some other Monococcus also. Today I'm going to mainly talk about these two algae. So what are the work we already performed in our lab and published in the scientific forum. Okay, so why carotenoids? So you know the carotenoids are the natural pigments produced by plants, algae, and also certain bacteria. There are about more than 1,100 types of carotenoids, and which are mainly classified into xanthophylls and terpenes. So these carotenoids have a very important role in plant as well as other organisms by involving in the photosynthesis and to capture the light and converting them into energy. So apart from plants, nowadays the plants are the major source, like tomato and other plants are the major source, but microalgae also is a great source of carotenoid because it can be able to grow in a smaller area and also will get large amount of yield. And also the time of harvesting also very less. When you see that tomato plant, at least it will take 2 to 3 months to harvest the fruits. Whereas here within 20 to 22 days, we can able to harvest the particular carotenoids from the algae. And as also the important role of carotenoids in plants and algae is, like as I mentioned, that involving in photosynthesis and other physiological functions. But in addition to it, for us, it gives a high commercial value in various industries like pharma, food, and cosmetic industries. So coming among all the algae like macro and microalgae, so microalgae is considered to be very good source of carotenoids. For instance, that the algae which we are going to talk about is the salina is produced about large quantity of carotenoids, about 100 gram per kg of dry weight. So today we can discuss about the important carotenoids like lutein, phytoin, and other total carotenoids, especially the beta carotene. In this talk, we can discuss what we did and what are the biotechnology we did in our lab. So first one about phytoin. So phytoin is a precursor molecule for the carotene, and it is a 40 carbon intermediate. That means it won't, it's not available in stable form. That means it will immediately convert into the other form. I think I will show the pathway, then you will understand. And so it has 3 conjugate double bonds, and then colorless carotenoid. So this is the important property for this carotene. And why, as I mentioned, is colorless carotenoid, because most of the cosmetic industries required this colorless carotenoid, other than the color carotenoid like carotene, beta carotene, or lycopene, all these things. That is colored. So normally, all the creams what we are want to purchase and use for cosmetic industry, we need a white color one. So that's why this compound is very, very important. And more than that is that it is more stable than other carotenoids. So compared to lycopene and other intermediates, it is more stable. And also it has very important properties to protect skin by UV absorber, and also involving in antioxidant and as well as anti-inflammatory. So these are the important properties of this phytoin. So industry people are harvesting this phytoin from the this particular algae I call that salina. So here, so how they're doing, there are many companies in India also. So even EID Perry also doing that, and Chennai. And these are the cosmetic companies like IBR, CLC. They are producing these colorless carotenoids, and these different products like IBR, CLC, IBR, AAC, IBR, TCLC, like colorless carotenoids, anti-aging carotenoids, tomato colorless carotenoids like phytoin, phytofluorine. Likewise, so you know that this all the phytoin is used in various products. So how they are doing, how they are isolating, as I mentioned that it won't stable or it won't stay for a long time in a phytoin form. Immediately when phytoin form, immediately converted into lycopene. Lycopene will be available in the longer in the plant or algae in the for longer duration. But what these companies are doing, they growing this algae for this life cycle for algae, or harvesting time for this algae is about 22 days to get the yield. So 16th or 17th day, what will they do? So once you get the enough biomass, when the algae are the green in color, I think you can saw the photograph in the first slide. So this is green in color. It will turn into red color within 2 or 3 days. So before turning into red color, because all what they'll do, they know that life cycle. So immediately they will add any one of these herbicides. What I given here, no, pyridoxine, difluronone, noroflorazone, or any one of these herbicides to block the enzyme which converts the phytoin. This is the pathway. So now you can see the pathway like say DM IPP or DMAP is converted into GGPP, general pyphosphate, and this general pyphosphate is converted into phytoin and to beta-carotene and to lycopene, and further the branches into 2 pathways as I mentioned already, like carotene pathway, xanthophyll pathway, and again one areas go to beta-carotene, zeaxanthin, anthocyanin, vioxanthin. Likewise goes to xanthophylls, and other areas like alpha carotene and lutein is goes in this way. But our aim is here to isolate the phytoin. This is the first product and intermediate product. It won't stable for longer time in the plant. Immediately, with the help of phytoin desaturase, it converted into beta-carotene and followed by lycopene. This is available. This is the pathway in the plant. Whereas when you see the same pathway in microbes, here there are 2 enzymes involved in converting phytoin into lycopene, is PDS and ZTS. Whereas in microbes, there are only one enzyme, CRTL. So it converts phytoin directly into lycopene. So that's you may have studied golden rice. This is the pathway which introduced in the golden rice to produce beta carotene. What they did, they introduced PS5, phytoin synthase, and then PDS, phytoin desaturase, and CYCB, lycopene cyclase B. these three genes are introduced to produce the beta carotene in the rice seeds. So this is important pathway in, and also not only this, this pathway branches differently, producing abscisic acid here I shown in this slide. But whereas there are two more growth hormones also produced in this pathway like GATN and GA3. But I didn't show that. This is branching into different other pathway. That is in an upstream way, not in this region. So you know that it is a very important pathway, like able to produce three important growth hormones like ABA, GA3, and cytokinin S-GATN. And so here, what our the company doing is that they want to isolate the phytoin by blocking the PDS. So to block the PDS, they added the many herbicides or some chemicals in the field. So what we thought, so why we want to pollute the environment, why we want to add. So when you when you're able to have a genetically modified strain of Dronella salina, that can be used to accumulate. That means we can able to isolate the phytoin. So what we thought is we can inhibit the PDS by using two different technology like RNA silencing method as well as antisense mechanism. That is our goal. So we wrote this project proposal and submitted to CSIR, and also they granted and we performed it. So our aim is like accumulating the phytoin by inhibiting the phytoin desaturase. That is the so we can able to isolate the phytoin without any adding any other chemicals or herbicides, and also it won't contaminate the environment. That is our purpose. Okay, so first, so we got this strain, Dronella salina, and this strains are growing in D valence medium, and the culture conditions is like 20 degrees Celsius and pH 8, salinity 12%, less density is about 2000 lux. So this is the how we maintain them in the liquid culture as well as in the agar slants. Okay. Once we have this, so we have to go for isolate the gene by two different methodology. For that, we need the PDS, phytoin desaturase gene. So we isolated the gene by isolating RNA and converting into cDNA, and from that, so here, so when you want to overexpress, we require the complete gene of interest with codings from start codon to stop codon. Whereas here we are only going for a gene silencing by two different approach. So we need only the partial fragment. That is the only partial fragment is sufficient for here. So we isolated the partial fragment and confound by gene sequencing, and also we found out that it is a unique one. It is not matching with any other already existing PDF gene in the database. So we got the partial gene sequence, and we cloned it into p-enter vector, topocloning vector, and then further we have to transfer into two different vectors, as I mentioned. One is to antisense vector. So the antisense vector, we have to clone our gene of interest in two different orientation to make the double strand RNA in the cell. Whereas other one is the PMDC45 vector is cloning in the antisense orientation. RNA vector, we need to clone in both sense and antisense, whereas in PMDC45 vector is the antisense vector, we have to clone our PDS in the antisense orientation. So now we first we conformed our transgene with the p-entry vector. So here we use the topocloning, that gateway cloning technology. Without digestion, we can able to transfer a fragment from one to other vector. So in such way, we have the region like ATTLR region is available. So this is the region will be recognized by the recombinase enzyme. It will transfer the fragment from one to other vector. So now we have two different vectors: Panda vector for the RNA construct, and PMDC45 for containing the antisense construct. So these are confounded by PCR as well as again by enzyme digestion, and we have the insert now. So now from this, so as I mentioned that the gene was transferred into LR clonase reaction by using gateway clone technology, and again after that also we conform by the resistant digestion as well as the colonipacia. And further, it is transformed into agrobacterium strain by using Phase3 method and also conformed. But here, before choosing the agrobacterium strain, so this is we searched literature and found out there is a less study or less transformation in the Dronella salina. So we have one study from CFTRI Mysore. They also worked on it, but they only studied by using glass bead methods. So we don't know which strain have to use, because we have a lot of reports are planned, because this agrobacterium transformation is well established protocol and only reliable protocol for stable transformation. and a lot of reports available for plant. But for this Donnelly algae, we have only few reports that are not stable transformation. They all are reported only this temporary transformation, especially for this Donnelly salina. The CFR report shows only temporary transformation by using glass bead method. So other methods like we have like electroporation and so you have to use electrophorator. This is available in our lab. And biologistic, so need this specific instrument, so it's called as the particle bombardment or gene gun method. That is not available in our lab, so we cannot use that. And silicon carbide whiskers and other method like protoflush transformation, microinjection, and PEG method is like again coming under the classification of protoflush transformation. These are different methods to get, but all these things what will do, it won't give the stable transformation because the whatever vector we will go, it will remain inside the cytoplasm, will express with the help of the algal machinery, the algal enzymes and other thing, and will go show the results. But for getting the stable transformation and to integrate our genome of interest inside the genome of the algae, we have to go for the agrobacterium transformation. So so before that, I want to introduce how this agrobacterium transformation confers the integration of this trans inside the plant cell or algal cell. So this is the summary of the agrobacterium transformation, as all of you know. So agrobacterium causes disease called crown gall disease. So it in the near to the crown region, that is the near to the soil region, the plant will get some disease called crown gall disease because of the tumor formed inside the plant. But that again, tumor is induced by the agrobacterium. That's why we call it the TA plasmid. TA stands for tumor-induced plasmid. So why is agrobacterium causing disease to the plant? So agrobacterium is solibacterium. So which required food is the opine. So what it does, it transfers the piece of DNA inside the cell. The piece of DNA contains three important coding genes. One for coding for auxin, other one coding for cytokinin, other one coding for opine. This opine is the food for the agrobacterium. But when this the tDNA goes inside the plant cell, so here again there are a lot of steps takes place here. Like whenever the plant cells are wounded by the insect's bite or any other natural happenings, or what happened, the plant cells will release the cell solutes. This cell solutes are sensed by the agrobacterium. So here the tDNA is the DNA which enters inside the cell, and as well as the other important region is the virulence region. Vir region we call it. So this virulence region produces a lot of proteins, like example vir A, B, C, D, E, F, G and all. So these are the very important protein which these proteins are playing an important role in processing or copying the tDNA and transporting them inside the plant cell as well as to inside the nucleus, and also involving integrating them inside the nuclear genome. So this is not happening in other methods, as I mentioned, like glass bead method, and all. Just we can shoot the DNA inside the cell. That's all. So it cannot cross the membrane like nuclear pore and go integrate. Sometimes it may spontaneously takes place, but 100% is not sure. So all other methods like protoflush transformation and electrophoration, what we are doing here, we're just transferring the DNA inside the cell, but it won't reach the nucleus because it have to cross the nuclear pore. To crossing that, the gene required the nuclear localization signal that is not available here. So and also we will transfer the DNA not in a linear form, because linear form DNA will be again digested or cleaved by the nucleus system, especially the endonuclease system. So it will chop the DNA or exonuclease will chop from the 5' end or 3' end. So it is we won't transfer in linear form. Always we transfer DNA in the plasmid form. What we'll do, we will clone our gene of interest in the plasmid and transfer it inside the plant cell. So all these cases, what happened, it won't reach the nucleus. But agrobacterium, so here it copies only the tDNA region. So this tDNA in originally it contains the auxin coding gene, cytokinin coding gene, and then opine coding gene. So here all this is taking place with the help of the virulence protein. So vir A is initially activated by the plant solute leakage, the chemicals, mainly the phenolic compounds or sugars or other thing, which will activate the vir A. And vir A will activate all other vir proteins, including vir G. So vir G again activates, go on bind to the enhancer elements and the promoter region of all other vir proteins and produces the vir C, D, E, all the things. So now, so where D2 will go on bind to the TA plasmid region, recognize the right border sequence and makes the nick. So nick means it cutting the only single strand, not double strand, and it copies the tDNA from right border to left border. And when it reaches the left border, it stops a nick again and releases the single strand DNA. And the single strand DNA will covered by where is taken by D2 protein as a pilot protein, and it again covered by where E2 by 600 molecules of E2 protein, and is ready to transform. So this is called as a T complex. The T complex is made up of one molecule of the D2 and 600 molecule of the E2 and one copy of tDNA. So this will be transferred into the plant cell or the algal cell. How it contains it makes a by through the conjugation tube or the transfer structure like tube-like structure is made up of Vir B proteins and Vir D4 proteins. So Vir D4 and Vir B we have 1 to 11. So 11 plus 12 D4 protein 12 proteins will form a channel or tube-like structure which help to cross the tDNA from agrobacterium to plant cell. So again, Vir B also here plays a major role. So here after coming inside the plant cell, it is coming to the cytoplasm. So it have to again cross the nuclear pore and have to integrate inside the nucleus. So for this here again, so this Vir D2 and Vir E2 contains nuclear localization signal. With the help of that, what can be able to cross the nuclear membrane, and finally it carries the DNA inside the into the nucleus. So this is how we are utilizing the help of agrobacterium, especially the virulence proteins function. So based on that, we can able to transfer it. So what we are doing now, we are removing the oncogenes. The oncogenes are the oxygen coding genes as well as cytokinin coding genes. We are removing as well as open coding genes, and we are placing our gene of interest, the transgene, the tDNA, as well as one more gene for the plant selection. So because we know we have to screen the plant for transgenic, for that we need one marker also. So we are putting that and we are transforming. So that's why we are using agrobacterium transformation for our carrying the transporting the transgene inside the plant cell or the algal cells. So here, so that is normal process when the plant cells wounded it release. But here we are doing experiment and we want to transform the gene from the agrobacterium to the algal cell. But here, how we can induce the virulent genes? There are many compounds, as I mentioned, that the phenolic compounds are like acetylsalicylate, cinnamic acid, vanillin, coumarin, glycine. These are the some of the different compounds are already established or reported that this induces the agrobacterium to produce the virulent genes, and the efficiency will be the transmission efficiency will be very high. But when we use this study, there is no difference in the level of transformation. So we want to include some other compounds, like there are some other reports also, like adding the sugar molecule, the glucose molecule. So this virulence protein, this phenolic compounds is regulating the virulent protein only. But whereas this D-glucose, so we added this D-glucose in the medium to induce the virulence genes. For a transformation of tDNA from agrobacterium to plant cell in the cells. So not only the TA plasmid, it also required the role of agrobacterium chromosomal genes also. So here the CHVE is one of the gene which will bind to this glucose. So the glucose binding protein is there. So this again activates the Vir A. So it's not directly the phenolic compound directly activating the Vir A, but here activating the CHVE, and the CHVE activate the Vir A and Vir A activate the Vir G and other or regulation course. That's why we added. So based on the literature in plants and other systems, we, along with the acetylsalicylate, the phenolic compound, we also added the D-glucose to induce the virulence system. And also know that there are many agrobacterium strains. In plants, there are already reports are available, but in algae, there is no report, especially in Donis arena. So we tried with 3 different algal strains, GB3101, LBA 4404, and EHA105. And also you know that each of them having a different chromosomal background, and also they are also having the different type of opine producers also. So, and also all of them coming under by using if I'm we can able to select the algae, this one agrobacterium. So what we did, we took this. We want to confirm the best agrobacterium strains among the three. So we got the strains and we transformed the transgene and PMDC vector alone, and we confirmed the transformation. So here again, so we tried with different strategies, like by using like control without association decomposed and only association and only decomposed, and again different concentration of decomposed, because we know that association is required for 100 micromolar. So we used a standard one and we get altered the different concentration of decomposed 5, 10 and 15 millimolar. And after that we co-cultivated and we study the effects in the all the. So here before co-cultivating, one important thing parameter I want to discuss, because algae is the marine algae, it requires the salt concentration, whereas agrobacterium does not require, no need of salt to grow. So now, so if you inoculate co-cultivate both of them in a single medium, it will be difficult to, algae cannot grow because it requires salt. So we have to standardize or acclimatize the algae and agrobacterium in a common medium. So we have to so we have used the tap medium here to co-cultivate. So slowly we increases the concentration of. So here you can see that. Okay. So so what we did, so we co-cultivated both agrobacterium and this algae in the tap medium, and then for about 48 hours we co-cultivated. And after that, we removed all the algae by washing, cephaloxamine washing, and then we inoculated the algae and subcultured and find out the transformants here. So here we use only the PMDC45 as a transgene. So here after transformation efficiency, we calculated and we studied the expression of GFP, because this gene contains this vector contains GFP to found out the transformants expression. We analyzed GFP gene, and for transgenic copy number of transgenic, we did PCR as well as Southern blot to analyze the copy number of the genes also. So this is the like antibiotic sensitivity test, because no, we have the antibiotic marker hygromycin in the vector, so we can able to grow in the transgenic algae will able to grow in the hygromycin medium also. So how can decide? So we have to concentrate before going to selecting the concentration of the hygromycin. We checked with the hygromycin concentration, and we found out that 3 mg per liter will be sufficient for going for the hygromycin concentration. And again for cephaloxamine, as I mentioned that you have to standardize the protocol for how much amount you have to use it here. And agrobacterium, so will survive even 100% of the cephaloxamine will survive 36%. So we have to find out, we have to use 300 microgram mg per liter. So that's when the agrobacterium will not survive. But again, we have to show that cephaloxamine should not kill the algae. So that also we searched here, and we found out that it won't killing that. So we can use 500 mg per liter cephaloxamine to get only the transformants, and it won't kill the Dunlalia, but it will kill the agrobacterium. So why we have to eliminate the agrobacterium? Because after transformants, because Dunlalia and agrobacterium, we have to recover the recombinant molecule, but both are having the recombinant plasmid inside the cells. But if agrobacterium are not removed properly, what happens? Agrobacterium will grow very fast and it not able to grow the Dunlalia. So we have to remove completely agrobacterium. So for that we are standardizing protocol with the help of the cephaloxamine and other antibiotics. Okay. So once we finalize the concentration of cephaloxamine and other antibiotics, and now we can able to see the transformation efficiency and the different strains. So you can see that efficiency was higher when we are using a azoxystrobin plus 10 millimolar glucose. So also in all agrobacterium strains, even in the HA105 and GB3101, also LBA4404, you can see that compared to the azoxystrobin alone or glucose alone, so the 10 millimolar concentration gives the good efficiency, and also you can see the same the colonies grown in different tap medium. Also you can able to see in the so like. A is the AHA105, B is the GB3101, C is the LBA4404, and D is the wild type cells that does not contains any transgenic. So because we are adding hygromycin here, so the wild type cells will not grow in the hygromycin. And again, we confirm the transgene, presence of transgene inside the, so we know that this is we screen them by using a selective antibiotic. But again, we should know whether this algae contains the transgene or not. For that, as I mentioned, that we have the GFP gene. We designed the primer using GFP and as well as HPT. HPT is hygromycin phosphotransferase, that is the coding for hygromycin antibiotic marker. So we designed the primer including GFP as well as HPT, and isolated the genomic DNA and amplified both GFP and HP gene, and we can able to find out if gene is present in all the transgenic lines and not in the wild type plant, because wild type does not have the transgene. And further, we want to confirm the copy number of the transgene. As you know that how many copies each algase contains. So we selected a few lines in each agrobacterium strain, like LBA44, we have about 6 lines, and we selected each 6 lines from each of the agrobacterium, and found out that LBA4404 gives most of them are 2 copy number. You can see there are 2 bands in the southern lot, whereas GB3101 gives 1 copy number, as well as AHA105 also given 1 copy number. So based on this, to be able to confirm. So like this is, we have just shown only for 6 lines, but we have different 18 transgenic lines. We analyzed it and we found out the transgenic the copy number 2 copies, and so you know the all LBA44 given 16 among 19 lines, 16 lines gives 2 copies, and 2 lines give a single copy, and 1 line doesn't give any results. So likewise, we can see that table for different agrobacterium strains for their transgenic efficiency as well as the copy number in the genome. So again, we again for contamination, we again studied for transformants for after some time whether any contamination is there, but if there is no contamination, because sometimes agrobacterium contamination also, when you isolate the genomic DNA, also will give the other positive results in PCR. But here there is no bacterial contamination. We got only the transgenic algae only. And also integration or stable integration also be analyzed, and we also found out that all the lines contain the transgene. Okay, so from this study, what we are concluding is that we evaluated 3 different strains, and with the help of pre-induction conditions like testosterone in the presence, deglucose in the presence of testosterone, and the efficiency was high in LBA4404, followed by GB3101, and followed by EHA105. But as you know that the agrobacterium differs not only in the efficiency, also is the copy number. So what we did, we took GB3101 because we need a single copy lines for further process to get any information, or we want to study further. So what kind of study we have to do is like so that the transgene inserts in a intron region, or it inserts or it inactivates some of the genes. If you have multiple copy genes, we cannot multiple transformation genes, it will be difficult for us to predict that. So if it is only one site, so easily we can find out in which site it was integrated. So we need the single copy integration site will be very useful for us to further study. That's why we selected GB3101 for further study. So coming back, so this results like synergistic action of deglucose and testosterone agrobacterium strains for efficient RNA transformation, we published in PLAS 1. So now based on this results, so we choose GB3101 for transferring the original study which we want to do for blocking the PDS by using RNAi as well as antisense vectors. So Panda is the RNA vector and PMDC45 is the antisense vector. So now, so we selected GB3101, coming back to the main story, and we co-cultivated, as already we standardized the protocol, and we got the 17 PDS RNA lines and 15 PDS antisense lines. So all the protocol are what same what we discussed earlier for the PMDC45 with only GFP, when we go on for standardizing the transformation efficiency with different agrobacterium strains. So now, okay, so we had again confirmed, so we isolated the genomic DNA from all the lines, so 17 RNA lines and 15 antisense lines, and even though it grown very well in the antigenic, you can see that it is the different line growing in the medium. But when you analyze the transgene, so we can find only among 17 lines, we got only 6 lines contain the transgene. Again, among 15 lines, only 5 lines we got the transgene. So for these lines are first selected and also analyze the copy number. So interestingly, the same results we got only single copy number in all RNA transgenic lines as well as the antisense lines. So now we took all these lines for the further study. So initially we did the RT-PCR analysis to see the gene expression study. So what we did, we blocked the phytoin desaturase. So we want to see what are the effects of for the other gene. So see, we analyzed this in both RNA line as well as antisense line. So you can see that the phytoin synthesis is precursor one. So this is the because blocking that, what happened, it accumulates the phytoin synthase, so it produces poor amount of phytoin. So you can see there is accumulation with the when you see with the control, and accumulation was high in the transgenic line number 5 in the RNAi, whereas there is no significant difference in accumulation of different transgenic lines in antisense technology. Whereas coming to blocking that phytoin desaturase, this is gene we blocked by both antisense as well as RNAi. But RNAi shows very good amount of blocking, significant decrease in the gene expression, whereas antisense we have decreased, but there is not much amount of decrease in the expression of this particular phytoin desaturase. And lycopene cyclase and the beta-carotene hydroxylase, these are the downstream genes in the same pathway. Here also there was decrease in this because we know that there is no intermediate product to convert, there is no accumulation here. So, but there is significant reduction in the RNA lines, whereas there is a reduction in antisense lines, but this is not compared to the RNA lines. So we have a good result in antisense RNA lines. Again, this level of, so again, that is the previous data is for about gene expression. This is about the content of phytoin. So you can see that when you measured it here, we can control, as you know, that control is immediately converted into lycopene, whereas there is we're not able to measure the phytoin content. Whereas here in the transgenic lines, you can see that there is a good amount of accumulation of phytoin in the transgenic line number 5 compared to our lines in RNA lines. Whereas antisense lines also we have a good amount of accumulation of this phytoin. So coming to the other carotenes like beta carotene, you know, because this is we don't have the intermediate because we don't have precursor phytoin to convert into lycopene further to beta carotene, so the amounts are decreased here. And also lycopene also there is a decrease in the amount of especially the line number 5 because it has less amount of lycopene and beta carotene. And also lutine also, I think same level of decrease was observed in, mainly decrease was observed in RNA lines and compared to the antisense lines. And also growth and other changes also is not up to the level when compared to the transgenic lines, because transgenic lines you can see the good growth and biomass are observed, and also chlorophyll control also very good in control plant. But when you compare with the transgenic lines, the chlorophyll control also decreased. So there is growth is due to the low photosynthetic efficiency compared to the control. So why this is because you know that so when we grow all this line antisense line as well as RNA lines with the high light condition, so you can see that the algae not able to grow because the algae is not able to produce the different kinds of carotenoids, especially lycopene, beta carotene, other things, lutein and all. So it not able to withstand the high light condition. So here, as I already mentioned in the introduction, the carotenoids are components for the assembly of photosynthetic apparatus of green plants. So what is doing? It serves as accessory pigment and light harvesting and play active role in electron transfer process of photosynthesis too. And also the essential function of carotenoid to protect the photosynthesis against photooxidation caused by the excited double state of chlorophyll. These are the roles of these pigments inside the plant cell, especially harvesting the light and also involving in electron transport chain, and also protecting them from the photooxidation. But here, when we block the production of the phyto beta carotene and other things, so we are only accumulating phytoin. We are not able to. We are reducing the amount of phytoin, lycopene and other carotenoids. What happened is due to the chlorophyll degradation because of the high light effect, or it's called as protobleach effect. So, so it not able to complete the cyclic the formation of because of the decrease in the photosynthesis of because of that decrease in the chlorophyll content as well as the growth, all the things we're not able to. Again, all this caused by because of the high light conditions. But when we grow them in the natural light, it able to grow in the laboratory conditions. But whereas you can see this diagram, the low light conditions able to grow. But whereas when you go for high light, it's not able to grow in the high light condition because of the highlight. It will not damages the system. Okay, so what we are saying from this story, so we successfully developed the transformation technology for this donor salina, and also be able to accumulate phytoe in this donor cells by suppressing PDS. And the reduction of PDS was high in RNA lines when compared to the antisense lines. And the line number 5 showed maximum content up to 108 microgram per 100 mg of dry content. And as I mentioned that partial inhibition of PDS gene leads to chlorophyll degradation and leaching effect causes the malfunction of photosynthesis, especially in phytoin accumulating cells under high light conditions. So not only that, the current study revealed that PDS functions as the major control point for the carotenoid synthesis and also for photosynthesis. This is our finding from the study. So this we published in Biophysics Technology on the topic like accumulation of phytoin, a colorless carotenoid by inhibiting of inhibition of phytoin synthase in donor salina. Okay, so now so we didn't expect that the high light will cause that kind of effects. So we change our strategy, like why don't we overexpress? So instead of blocking that, if you overexpress this phytoin synthase, that is the precursor or the gene which accumulate the phytoin or converting the GGPP into phytoin. So that again we wrote one more project like accumulating overexpression of phytoin synthase to accumulate more amount of phytoin as well as other compounds also. Again, this is funded by DBT. So the work is going on now. So we are overexpressing this PSY, not PDS, PSY, the precursor, the first enzyme involving this particular pathway is called as phytoin synthase. So for the enhanced production of carotenoids, not only phytoin but also all kind of carotenoids also. So this is, still we are working on it. We not say that result, but in addition to that, we also utilized various kind of stresses to induce these carotenoids in this algae. So for that, what we studied, we took the nutrient depletion studies along with supplement of sodium bicarbonate. So here we took 3 different nutrient stressors like nitrate deficient, and then phosphate deficient, and then sulfate deficient we took. And for and all 3 we also supplemented with sodium bicarbonate. So I think you can see there are 7 different graphs. One is the total nutrient, and then nitrate minus nutrient, and then phosphate minus nutrient, and sulfate minus nutrient, and also each of them supplemented with the sodium bicarbonate. So we want to see what will be the role. So why sodium bicarbonate? You know, the sodium bicarbonate is another form of carbon dioxide. So the carbon dioxide we cannot give. No, so to increase the efficiency of the photosynthesis or cell growth or biomass, so this will be enhanced. Even though when we depleting a particular nutrition will give stress to the plant, but this bicarbonate will enhance the growth or pH, all the things in the medium, so we'll be able to produce the higher contents. So this to overcome or tolerate the stress, the plant will produce a lot kind of carotenoid. So this is the plant again, it will mainly produce this kind of carotenoid, especially donor salina will produce beta carotene. So here you can see the graph shows the growth, pH and biomass, and you can see compared to the control, the stress conditions able to there is significant reduction in the growth also, but you can see that to compare to the nitrogen depletion, the supplement sodium carbonate able to recover the growth as well as the pH also. And I think you can see the data on protein lipid carbohydrate production. You can see total nutrient, we can see this less amount, whereas nitrogen depleted medium, you can see there is less amount of protein, but there is a good amount of lipid and carbohydrate. But when you supplement with sodium bicarbonate, you can see there was increase in lipid and carbohydrate content. So when you want to accumulate lipid in the algae, so I think it's best is you can go deplete the nitrate, and then we can supplement with sodium bicarbonate. So likewise, but there is no significant difference in phosphate depletion medium as well as sulfate depletion medium. Almost you can see the in all cases into control and other things, at most have the same. Well, there is slight reduction, but not significant then compared to the nitrate deficient medium. So main thing is like the important thing is like we have to see that the beta carotene, the carotenoid content in the donor salina, you can see they can compare the control. So when you have nitrate depletion medium, it's able to accumulate twofold, when you supplement with sodium bicarbonate, you can see it accumulate about 4-fold. So normally it's 50 microgram, whereas it goes up to 200 microgram. So the best method is like we can deplete the new nitrate as well as supplement with sodium bicarbonate. So this data we published in bicarbonate and the scientific reports. The title of bicarbonate supplementation enhances growth and biochemical composition of Donelli salina by replacing oxidative stress and induced during macronutrient deficient conditions. This is the how it increases the beta carotene. So coming to the next example, like lutein, is from other algae like Chlorella pycnosa. This is the marine, freshwater algae. So here, so we focused on beta carotene in Donelli salina, whereas here we focused on the lutein because this is a major producer for lutein. So again, you know, the lutein is the end product in this pathway. And you know, apart from algae, plants, especially marigold flower, gives lot amount of lutein. And you know, the importance of lutein is, you know, it is used just a food additive to give the good color, and also especially in the poultry field, like poultry feathers, and also give deep yellow in the egg yolk, and also many foods we are adding as the flavor and color to foods. And also other important functions like it's also useful to treatmenting the ameliorating cardiovascular diseases and then cancer treatment. And a very important thing is like age-related macular degeneration. So this like eye disease also we can able to control. So now we want to enhance the production of lutein inside this particular Chlorella pycnosa. So for this we taken different stress like salinity and nutrient starvation and sodium bicarbonate. So you can see that growth condition, you know, that when you, because this is a freshwater algae, so when you use this saline or salt treatment, you can see it able to grow well up to 60 millimolar, and after that 40 to 60 millimolar. After that 40 millimolar is fine, but after that, you know, the growth was reduced. And nitrogen starvation also. So as I mentioned that the nitrogen deposition medium always no, it decreases the growth, but whereas other phosphates another thing, it also reduces the growth, but not like to the like the nitrogen starvation. And sodium bicarbonate supplementation, as you know that it, and compared to the control, again here also concentration depends. Here again 100 millimolar gives a good growth of this algal cell. And chlorophyll content also is relevant to the same of the previous graph. So salinity, whenever we have the high salt concentration, the chlorophyll content A content reduces. Again, in nutrient starvation, especially nitrogen starvation, the chlorophyll content reduces. And also here the sodium bicarbonate supplementation also, when we have go the higher concentration, it reduces. So 100 millimolar gives a good amount of chlorophyll content, whereas after that there is a decrease in the chlorophyll A content. The similar to the chlorophyll B also. And total carotenoid, so it includes all the carotenoids, including whatever carotenoids are available in the chlorella, including lutein, beta carotene, other things. You can see that the salinity stress also 20 millimolar, it gives a good amount of total carotenoid, but after that, even the growth was good to 40 millimolar, but there is the reduction in the total carotenoid. Among the nutrition starvation, so the nitrogen starvation, even though growth was the less, but it able to accumulate high amount of carotenoids in the cell. So like similar to the Donella salina. And sodium bicarbonate supplementation also. So it able to accumulate when compared to the control, it able to accumulate a high amount of total carotenoid, but the amount was high in the minimum constant like 100 millimolar sodium bicarbonate. But when you increase the concentration, there is increase in the compared to control, but as not up to the mark to the 100 millimolar sodium bicarbonate concentration. And again, lipid production, the similar trend was repeated here also. Like in the 20 to 40 millimolar, we have a good amount of lipid content. And also you can see that we analyzed this content in a different growth stages, like in 5th day, 10th day, 15th day, 20th day, and 25th day, and 33. I think you can see that in almost all cases, the 20th day is the best treatment, best for us to harvest all these compounds. So total carotenoids or lipid content is best for us. And here we also check the lutein content, especially because this algae is mainly for the lutein content, and we analyze that in 20th day. You have the good amount of lutein expression was produced in the salt stress. So that stage we again analyzed for checking the expression of different genes like CHY gene, LY, phytoenzyme change, lycopene cyclase, and all the things we analyzed here. Here also you can see that at 40 millimolar the gene expression was high. So here we took only that 20th day for our experiment. We can do check for all other days. We took 20th day, and again it's a different concentration, control 20 millimolar, 40 millimolar, 60 millimolar, 80 millimolar, and we can find out the 40 millimolar condition. We can have a good amount of gene expression of all three genes is involving in the lycopene pathway. And also this nutrient starvation, HPLC, we can quantify the lutein content. So you can see that best is 25th day. We choose this 25th day for us analyzing these genes. And also we found out in nitrogen depletion medium, it has a good amount of accumulation of these genes, like all three genes are accumulated in higher concentration in the 25th day. And then this is the supplement of sodium bicarbonate. And here also we found that it is a good accumulation of lutein at 30th day. And this particular day was cells were taken for further for the gene analysis. And again here the 100 millimolar concentration gives the good amount of gene expression of the particular cells for all the three genes like CHY, LYC, and PDS. So again for functional studies, so you know that there are a lot of functions as we discussed in the introduction slide of about the chlorella pyanosa and about the different role. And we want to do some functional studies in our lab also to validate our data and to publish our data in a good journal. So we went for the biofilm property of this lutein. So we also analyzed by 4 different kind of assays like biofilm inhibition assay and exopolysaccharide degradation assay and cell surface hydrophobic inhibition assay and picosine degradation. These are all the assays involving and confirming the anti-biofilm property. So we took that also. And we found that our compound, that lutein, used a different concentrations like 3, 5, 10, 20 microgram. And also we use the standard lutein and as well as the on positive control. So based on that, we can see that the dose dependent manner, our biofilm inhibition was high, EPS degradation was high, and cell surface hybridization inhibition also high, and picosine degradation also is dose dependent manner, it was high when compared to the standard drug as well as to the standard lutein which we got from the Sigma. And this again further one more says confirms the biofilm formation. Here we biofilm formation we use Pseudomonas aeruginosa, and we can able to see by treating with different concentration of lutein we can able to control the biofilm formation in the slides. Further, we also analyze the 2 different genes involving in quorum sensing like LAS and RHL. So here you can see the control without treatment, you can see the gene was expressed high, whereas when you treating with the standard drug like azithromycin and the standard lutein, you can see that the expression of these 2 genes are reduced. But when also we treat with our lutein compound, also you can see that there was reduction in the gene expression of in the 220 letters lines. So this also we published in Micropathogenesis. Like we say that lutein, a potential anti-biofilm and anti-quorum sensing molecule from green microalgae, chlorella pyanosa. So, okay, before going to that, so what is take-home message and what you understand here? So we can also use biotechnological approach to enhance the production of different carotenoids, not only lutein, and also lycopene and as well as beta carotene. There are so many carotenoids available in different algae like Hematococcus. It higher produces of it produces lutein and higher concentration, and also there are many more algae which has accumulate different carotenoids. Again, it is useful for our various pharmaceutical purpose as well as also used as a food also. Okay. So what is future prospects? So how we can do that? So one is like we can optimize the media concentration by depleting nutrition, especially the nitrogen, or and also we can supplement with the sodium bicarbonate. This is one method. Or we can also trying with other methods. Now we are doing with like adding the different growth hormones like IAA, IBA, and other kind of growth hormones. Also like jasmonic acid, methyl jasmonate, and all the things we are studying now by giving different stress and how we can accumulate at a different time interval. So after getting enough biomass, can we add induce the stress? So with the studies, what we are shown all is that the stress induced in the first day itself. It's not like after we will grow that we grow the algae in the nitrogen depleted medium itself. But what we are thinking that we have to grow the algae in normal medium, and then after that we have to deplete the nitrate, maybe after 15 days or after 10th day. So we can see the effect whether it is accumulate more amount of carotenoids or not. But when you are giving stress in the initial stage itself, the algae gets lot of stress and not able to grow properly. Their growth and biomass are decreased, and even after decreasing, we'll get good amount of lycopene and other compounds. But when we grow for sufficient, that means you have to change the medium. That is the one important other parameter that we have to grow in normal media for first 2 weeks, and then the first 10 days, then we have to change into the stress medium. That we have to do. But it may they are also working on it. So whether we can go for a different intermediate, like 10th day, if we give the depletion, what will happen, or 15th day view, what will happen. Like this, we are going studying on. In addition to the different stresses also we're doing. So coming to the biotechnological part, as we are biotechnological student, so what we can do, so now there are new technologies are rising, like the CRISPR-Cas. So can we modulate the PDS gene or PSY gene, pyrogen synthase or pyrogen desaturase, to overproduce or to act for a high expression. So by now, that I think all of you heard about CRISPR-Cas9 and CRISPR-Cas12 is involving here, like without introducing a new gene, just modulating or eliminating or modifying some of the elements in this region, we can able to overproduce these genes, and finally it leads for the production of higher amount of carotenoids, especially the beta carotene or lycopene or lutein. So these are the different messages I want to pass on it. The future direction. So anyone want to study, these are the different aspects varying for different stresses, even we can also go for radiation stress and different light stress and other growth hormone stress we can study. And also we can couple them, all the stresses together, and finalize or found out the best, optimize them for best producers or best accumulation of these carotenoids. That we can, again, we are some of our PG students and PhD scholars are working on it. Maybe in after 2 or 3 years, we can able to answer this question what I discussed now. So at the end, I want to acknowledge the funding agency CSIR. The first project for the blocking the gene PDS, it was granted, and the second project DBT, which is granted for overexpressing the pathogen PSY gene. And the first projects were carried out by Dr. R. Srinivasan, and the lutein work was carried out by the Mr. Sharman. And the second project, like DBT, now Ashwini is working on it. Maybe she will give good results in future. We will discuss it later on with you. And also, I thank the VT University for giving the necessary facility to carry out this research. So with this, I complete the talk. And any questions?
Thank you, sir, for the elaborate presentation. So, dear participants, now the forum is open for discussion. If you have any queries, please do post in the chat box, or you can unmute yourself and discuss with sir. Sir, I hope there is no questions as well as your. If there is any queries, we will get back to you, sir. They may type in the chat box and they will post it. If there is any queries, we will contact to you with the queries. Sir, on behalf of the Department of Biotechnology, staff, students, and management and HOD, and we extend your heartfelt gratitude for accepting our invitation and giving such an valuable and detailed explanation, sir. Thank you so much on behalf of everyone.
Thank you, ma'am. Once again, thank all the organizers and the biotech department and the college for inviting me to deliver talk in this
And
August
also,
forum.
we are
Thank you.
very glad to know that you are the guide of Dr. Ajita. So it is a proud moment for Ajita too that you be here. So once
Yeah,
again, thank you so much.
yeah. Thanks, ma'am. Thank you.
Thank you. Thank you.
Yeah.
Dear participants, with the kind consent of the convener, we are winding up with the session 1 of day 4. Please do join us back at 11:30 for the session 2 with the same link provided. Thank you. Thank you, Ananda. Sir, you may kindly log off. Good morning, Madhukumar sir. Can you hear us?
Good morning, madam. Yes, I can hear you loud and clear. I hope I'm also audible and
Yes, sir.
visible.
You are audible. Sir,
Thank you.
another 2-3 minutes we will
We'll
start.
wait. We'll wait. Sure. Madam, are we good to go?
Yes, sir. Just a minute, sir. We will start.
Right, I'll wait for you.
So,
Good morning, sir.
Good morning, sir. Good morning.
Happy to see you, sir.
Thank you. My pleasure.
So we'll start the session.
Yeah, I'm ready. I'm good to go.
Yes, sir.
so just a minute, sir. I will
Sure.
start.